Hey everyone. Look, we all know about DNA, right? The famous blueprint for life. But what if I told you that blueprint is, well, it's just the beginning of the story? Today we're going way beyond it, into the dynamic, messy, incredible world that's actually running the show inside our bodies. So let's just jump right in with a big question. What if that blueprint for life is seriously incomplete? Imagine you have the architectural plans for a massive skyscraper, but you have no idea what the construction crew is actually doing inside minute by minute. That's kind of the problem we're talking about here. And this is the perfect way to picture it. On one side, you've got the genome, our DNA. That's the static, unchanging plan. But on the other side, that's the proteome. That is the actual bustling construction site with all the workers and machinery moving around, doing things in real time. That is where the action is. Now, to really get why this difference matters so much, we have to talk about something called the genome's great gap.
Okay, quick flashback to high school biology. You probably remember the central dogma, right? DNA makes RNA and RNA makes protein. It all sounds so simple, so linear, a nice, neat little assembly line. But here's the thing, biology is almost never that simple. It's way messier and, honestly, a lot more interesting.
Let's just look at the raw numbers for a second. The human genome, our entire genetic code, has about 20,000 genes. Now, when scientists first figured this out, they were kind of shocked. Just 20,000 to build an entire human being? It seemed way too low. And, well, their gut feeling was spot on. Because, get this, the number of different protein variants, the actual little machines doing all the work in our bodies, is estimated to be over 1 million. So, wait a minute. How do you possibly get from just 20,000 instructions to over 1 million unique functional parts? That, right there, is the great gap. And the answer, it turns out, is all about the proteins themselves. They're the ones really running the show. They are life's real workers. You know, there's a great line in the source material for this that just nails it. It says proteins are the machines, bricks, and mortar that actually execute the functions of life. They're not the plan, they're the action. They are the reality of your biology at any given moment. So how does this incredible explosion from 20,000 genes to 1 million proteins actually happen? Well, biology has a few tricks up its sleeve. A single gene can be cut and pasted in different ways. It's called alternative splicing to create multiple messages. Then, once a protein is made, it can be decorated with all these different chemical tags. Each new tag can completely change its job. It's like having one recipe for dough, but by adding different toppings and fillings, you can make a pizza, a calzone, or a cinnamon roll. Same starting point, but radically different results. Okay, so with all this mind-bending complexity, you can see how scientists have a real challenge on their hands. If you want to figure out what's really going on in a cell, you have to choose the right tool for the job. And that brings us to the world of omics. This chart breaks it down perfectly. Let's start with genomics, studying your DNA. It's mostly static. It tells you your potential, what could happen. Then there's transcriptomics, looking at RNA. That gives you a hint of activity, what the cell is maybe thinking about doing. But proteomics, studying the proteins, that's the real-time feed. It tells you what is actually happening right this second. And this is the absolute key takeaway. Just because a gene is on and making an RNA message, that gives you no guarantee that the final protein is actually there and doing its job. If you want the ground truth of health or disease, you have to look at the proteins. All right, so we've established that proteins are a huge deal. They're incredibly important and complex. But how in the world do you even begin to study them?
The answer is this remarkable piece of technology called mass spectrometry. You can basically think of it as an unbelievably sensitive and precise scale for molecules. It is the absolute workhorse of this entire field.
And here's a super simplified look at how it works. You take a sample, like a drop of blood, and you pull out all the proteins. Then you use molecular scissors to chop those long proteins into smaller, more manageable pieces called peptides. Next, you separate that complex mixture of peptides, and finally, you feed them into the mass spectrometer, which identifies and counts them one by one. I love this analogy for the two main ways scientists do this. The discovery approach is like casting a giant fishing net. You just throw it out there and see everything you can possibly catch. On the other hand, the targeted approach is like using a harpoon. You know exactly what protein you're looking for, and you go after that one target with incredible precision. So you take all this amazing technology, all this deep biological insight, and it's all pointing in one direction. The future isn't just about reading the blueprint anymore. The future is functional.
But getting there is not easy, far from it. This chart shows one of the biggest hurdles, the dynamic range challenge. The best analogy is trying to hear a single pin drop during a rock concert. In your blood, some proteins are so common they're like the screaming guitars and pounding drums. But the really important signals for disease, these tiny proteins called cytokines, they're the pin drop. Being able to hear that pin drop through all that noise is the massive challenge.
And it gets tougher. We're talking about differences in the amount of proteins by a factor of 10 billion. Plus, with DNA, if you don't have enough to see, you can just make more copies. You can't do that with proteins. What you have is what you get. And the amount of data these machines produce is just staggering. It takes immense computing power to make sense of it all. But, and this is the big but, the payoff for cracking these problems is nothing short of revolutionary. It's just like our source material says. By moving beyond the static genome, we gain the ability to see the cell in real time. So what does seeing life in real time actually get us? Well, this isn't just an academic exercise. We are talking about diagnostics that can detect diseases far, far earlier than we can now. We're talking about medicine that's truly personalized to your body's current state. And really, we're talking about a fundamentally deeper grasp of the machinery of life itself. And that's really the thought I want to leave you with. For decades, we've been studying the blueprint. But now, we're finally developing the tools to watch the builders at work live. So the question isn't just what we've learned, but what's next? What mysteries will we solve when we can finally watch life as it happens?